Okay, coming to next question. Which one of the following technique is used for the characterization of RNA? See a characterization of RNA. So in the biotechnology, we use the word characterization of RNA. Okay. So here Western blotting technique, Southern blotting technique, Northern blotting technique, and Eastern blotting technique. This is not present at all. So we have three techniques are there for DNA, RNA, and proteins. Okay. Especially Southern blotting technique and Western blotting technique both are useful, commonly useful in the biotechnology, and for the RNA purpose, Northern blotting technique is used. Okay, if anybody of you know about the Southern blotting technique and Western blotting technique, please comment what is where we use the Southern blotting technique and Western blotting technique. Comment. Choose the correct transport sequence for protein transport in a cell. Okay, protein transport in a cell. They're asking about ER, Golgi complex, plasma membrane, nucleus, Golgi complex, plasma membrane, Golgi complex, ER, plasma membrane, cytosol, nucleus, plasma membrane. So this is the formation of protein synthesis they are asking about. Okay, here the cell, this is nucleus. Okay, nucleus, and the nucleus ER is present, is called ER. Okay, on the ER, rough ER is there, rough ER. So rough ER is the with the ribosomes. So here not asking directly about the ribosomes, he asked about the ER. So ER also participates in the protein synthesis by using ribosomes. So here whatever the proteins are created in the ER, that's called rough ER. Okay, for modifications, that protein molecule enters into the Golgi complex, So after modifications, the matured protein comes outside and attached to the plasma membrane. So this is the sequence of proteins movement, transportation, okay, from the ER to the Golgi complex and then to the plasma membrane. So here Golgi complex, plasma membrane, this is the answer for this question. Next question, see. In gas liquid chromatography, GLC, there are large number of stationary phases available. Yes. While in high performance liquid chromatography, HPLC. So this is a question about the chromatography we never expected in the zoology question, but most important for the separation of the molecules based on their color. So here in the GLC, gas liquid chromatography, there are stationary phases are more. But coming to the HPLC, what is happening here? Wide range of liquid stationary phases are appear. So HPLC, high performance liquid chromatography, liquid is there. So in the liquid chromatography, what happened? Liquid stationary phase. So the liquid chromatography means that this is the stationary phase. So here in the HPLC, we use that they have wide range of liquid stationary phases, whereas the GLC, they have stationary phases. Large number of stationary phases are available. Coming to the next question. What is the structural and operational component of a mammalian nervous system? Nervous system made up of axon, dendron, neuron, brain. It's very simple question. Neuron is the answer. Yes, any doubts? Which human disease was the first to be eradicated by a program of vaccination? Very simple question. Vaccination started with the Edward Jenner, and here in India, in the world, the vaccination program also conducted to eradicate smallpox. Okay, and here smallpox also, one question is here, smallpox vaccination cannot give long-lasting immunity. No long-lasting immunity. Means once you have taken this vaccination for smallpox, there is no guarantee that second time it will not occur. Some cases are there. In the some cases, in crores, one case or two cases are we are seeing in India also. We observed that certain cases with even they are taking this smallpox vaccination also, certain places they are getting the second time. So the disease appeared second time also. That is why we can say there is no long lasting immunity for this smallpox vaccination. Juxtaglomerular cells in the kidney secretes. Juxtaglomerular cells in the kidney secretes here renin, aldosterone, vasopressin, epinephrine. Epinephrine we know very well, that is epinephrine from adrenaline, adrenal gland. This is the endocrine gland. Vasopressin, also called ADH hormone, antidiuretic hormone, and aldosterone. These hormones are there, but the juxtaglomerular cells, they secrete renin hormone. This renin hormone. One hormone is there in the nutrition topic we discussed already, that is renin. See here, renin and renin, both are just single N different. Double N, renin, single N, renin. Renin from the juxtamolecular cells of the kidney secretes. Juxta means deep into the medulla. Kidneys are with two portions in kidney. So outer portion is called cortex, inner is called medulla. So there is a medullary or juxtaglomerular cells means here deep into the medulla. That's called juxtaglomerular cells in the kidney secretes renin enzyme. So this is a renin enzyme that converts the angiotensinogen 1 to angiotensinogen 2. So they convert that reaction. So most important, and a heartbeat also they control this renin. Which one of the following is not a vestigial organ in human beings? Very simple question, no need to any explanation, very simple. Appendix is the one of the vestigial organ in our body. Appendix, coccyx. So after the vertebral column, the ending part is there, that's called coccyx, also one of the vestigial organ. Pinna, ear pinna, also one of the vestigial organ. But in the teeth, so very simple question, can easily without any wasting of our time we can answer that question. So teeth are the not vestigial organ in our body. Mechanism of regulation between the entities and environment of solutions. The loss and gain of water is known as loss and gain of water. Suppose this is amoeba, this is water outside, and inside there is a cytoplasm, nucleus, cytoplasm, and these are contractile vacuoles, suppose. Okay, what happening here? Mechanism of regulation between the entities and its environment of solutions. The loss and gain of water. So entry of the water and the loss of the water. So this is gaining water, losing water, loss, minus means loss. So water loss or gain, that is known as osmoregulation. It's called osmoregulation. What is meaning of homeostasis and homostasis? See here both with thermoregulation. Thermo means here temperature. So this is completely wrong answer, not given. And homeostasis means here water balance in the body. Water balance. So balance is different from the loss and gain of water. So best option is the osmoregulation is the best option in the given 4 options. The role of calcium in muscle contraction is calcium. Calcium in the muscle contraction. In the muscle contraction, if you take the skeletal muscle, if you take a skeletal muscle fiber, skeletal muscle fiber is with alternatively present A band, I band, and A band, I band, like this. Okay. So here there is a T tubule. T tubule is present. T tubule is with calcium channels. Okay. Cistern, they have the bags are present. So T tubule is the bag of calcium, and whenever there is a signal from the T tubule to contract from the neurotransmitter, then immediately calcium ions enters into the cytoplasm and they go and binds with the troponin. So calcium binding with troponin, and then the mask of the troponin removed and exposes the myosin head to the actin filament. So this is the one of the reaction where contraction is the starting point for the contraction. So releasing of the calcium ions is the first step, where that calcium ions go and binds with the troponin on the actin filaments. Three sites are there: TNI, TNC, TNT. Troponin C is called TNC site. On the TNC, C for calcium binding site. Whenever the calcium from this T tubule cistern ER, they comes outside, and immediately they go and binds the TNC site of the actin, and then they release the active site to bind that myosin and come and binds with actin. So here, to bind with troponin, changing shape so that actin filaments are exposed. Yes, the mask is removed. So mask removed. That is the work of calcium. Yes, that is the answer. Second option is the answer. This one answer. Next, quadrate bone. See here, quadrate bone in the reptiles evolved to form which bone in the mammals? Okay. So malleus, incus, stapes. These three bones are present in the middle ear of the mammals. So malleus, incus, stapes. And coming to hyoid bone in the throat region. So hyoid bone is the one of the bone came from the or evolved from the quadrate bone of the reptiles. So here hyoid bone is the answer for quadrate bone in the reptiles, converted into here hyoid bone in the mammals. Which one of the following pathway is common to both fermentation and cellular respiration? So it's a very easy question for the respiration they are asking here. Fermentation is one type of anaerobic respiration. Anaerobic respiration. So in the anaerobic respiration, low amount of oxygen, or in the absence of oxygen, if yeast cells undergo fermentation and release the ethyl alcohol. This is the concept for fermentation. And cellular respiration we discuss in the aerobic respiration. Aerobic respiration. The respiration topic we are discussing every day, 9 p.m. to 10 p.m. 1 hour live session is there in the Zoology Online Guru YouTube channel. So come and join this channel every day, 9 p.m. to 10 p.m. Already one complete topic is completed. That is, the human digestive system is completed. So each and every point we covered in the human digestive system, go through those videos in the YouTube channel Zoology Online Guru. And already we started a live session about the human respiratory system. In that system, we are going to discuss all these things of the aerobic and anaerobic respiration. So kindly go through this YouTube channel Zoology Online Guru. and come and join the session at 9 p.m. to 10 p.m. 1 hour daily live session. So here aerobic respiration and anaerobic respiration, whatever it may be, in the both the respirations, nucleus, this is the cytoplasm, glucose from the glucose through the blood enters into the cytoplasm. Glucose breaks into pyruvic acid. And this pyruvic acid enters into mitochondria. So if it enters into mitochondria and oxygen is utilized, that is known as aerobic respiration. Glucose breakdown and conversion of the pyruvic acid or ethyl alcohol, it is known as fermentation. So glucose breakdown and formation of ethyl alcohol, otherwise glucose breakdown and formation of ATP by using the mitochondria, whatever it may be, the first step is to breakdown of glucose. So the option is glycolysis. Krebs cycle. No, wrong answer here. This is a Krebs cycle takes place in the mitochondria. Mitochondria means we are going to cellular, that means we are using the oxygen. So that is glycolysis. This is wrong answer. See, in the master question paper also they are with two wrong answers, they gave two wrong answers. Just now we discussed the first question. So here glycolysis, if you know the answer, comment. See here, we know very well that is glycolysis is the first step. Even it is aerobic or anaerobic, the first step is glycolysis, not the Krebs cycle. Without glycolysis, how Krebs cycle takes place? So in the master question paper, they gave this Krebs cycle. A is the option, but glycolysis is the correct answer. Electron transport chain, acetyl-CoA formation from the pyruvate. So formation of pyruvate is the first step. So if you know the answer, if my answer is from my side, my answer is the second one, glycolysis. From your side, if you are thinking about the another Krebs cycle is the answer, support your answer and comment. Please comment. Next, what are the causes and treatment of Addison's disease? Addison's disease is one of the autoimmune disease. It is autoimmune disease, Addison's disease. Here, in an inability to produce aldosterone, take in excess sodium. So aldosterone production is altered and to take excess sodium, that is why it's called Addison's disease. Is autoimmune disease. If you know about this Addison's disease, please comment about the Addison's disease. So option 4 is the correct answer only. So an inability to produce aldosterone, take in excess sodium. Next one, the science of studying and treating the malformation and monstrosities of organism is called monstrosities. Okay, this is different one. We know about the gerontology. We know gerontology becoming the old, okay, aged, that is called gerontology. Dermatology, skin, we know. Etiology also we know, but here teratology is the new one. Here the science of studying and treating malformation and monstrosities of organism is called teratology. This is the different one, very important. Okay, very nice question. So teratology also they asked in the MSc entrance zoology examinations, teratology. A water body is termed as eutrophicated. Okay, this is the eutrophication. So eutrophicated means here highly highly nourished or nutrients rich medium is called eutrophicated. So algal growth is more in that eutrophicated water bodies because an extra nutrients nitrogen or phosphorus nitrates or phosphates are adding to that water body and increasing the nutrient rich in that water body, and immediately the alga come and grow. So the greenery patch is appear on the water body. That's called eutrophication. So answer here, nitrates and phosphates increases that nutrient value of that water and creates the eutrophication. Option 3 is the correct answer. Yes. Calcium is important in skeletal muscle contraction. Yes, just now we discussed that calcium is binding to the TNC and it is opening the mask on the actin filaments and make easy to lock and key attachment of the myosin head to the actin, and then pulling of the, that's called contraction, takes place. Here binding to troponin TNC and to remove the masking of active site on the actin for meiosis. Same question asked once again, but type of question, the pattern is different, but question is same. So this if you know that just before we discussed the same question, so if you know that answer, this question also very easy to answer. Same answers are there. So troponin is with the TNC, and that C for calcium. Calcium come and binds with troponin and removes the mask and opens the active site to bind with the myosin to actin. So all these things not necessary. So this is the first one is also easy. That is answer. And here, which of the following is the correct sequence of prophase 1? Prophase 1 in the meiosis. In the meiosis 1. Prophase 1, there are 5 stages are there. There are 5 stages: leptotin, zygotin, pachytin, diplotin, diakinesis. So we have to arrange the correct sequence. First one leptotin, zygotin, third one pachytin. Pachytin is where recombination takes place. Recombination. That's called chiasma. So crossing over takes place. Crossing over takes place. That is why new characters arises in the offsprings by crossing over. So that's pachytin in the middle and diplotin and diakinesis. These are the sequence. So this is correct sequence. Second option is the correct one. Matching. Type of cancer. Okay. Carcinoma, epithelial cells. Chondrology, cartilage, chondrology, chondritis. So cartilage. Sarcoma, tissue. Okay, it's called sarcode. Is called muscles. Myeloma. See here, A4B2, C1. Okay, C1 sarcoma muscle. Myeloma, myeloid cells, blood. Yes, correct. Okay, first option is correct. Okay, A4B2C1D3. First option correct. Any species that have been accidentally or deliberately transferred from their original habitat to a new habitat whose abiotic conditions are also suitable for them are known as alien species or invasive species. So here alien species means they are transferred from the other area to this area. So the local species are disturbed by the alien species, because alien species, if they are transferred to other area, they may have the capacity to live at that new space, because the abiotic factors are favorable to that organism. That is called alien species. So local species are affected by this alien species. Next, so option A is correct one. Question number 31. In which of the following national parks Indian rhinoceros, okay, rhinoceros, Kaziranga, Assam. Okay, Kaziranga National Park, Assam is famous for Indian rhinoceros. Jim Corbett National Park, it is present in the Uttarakhand. Uttarakhand, the first national park in India. First national park in India. Jim Corbett National Park. Jim Corbett National Park. Manas, Assam. Manasa National Park and Kaimur National Park. And comment me where is the Kaimur National Park. If you know the answer about this one, Kaimur National Park, where it is and which animal is present in that park. Comment me if you know the answer. Comment me. Okay. Nucleosides are the compounds in which nitrogen bases, that is called purines and pyrimidines, are conjugated to the pentose sugar, yes, ribose or deoxyribose, DNA or RNA, by a bond. You have to know about the backbone of the DNA. So try to draw, we will try to draw a backbone of DNA. A adenine with a double bond with a thymine. Okay, G C, T with A and C with G. This is the sequence. This is the adenine. Here sugar, and also sugar. Okay, 5 dash and 3 dash and 5 dash 3 dash 5 dash 3 dash 5 dash 3 dash. So we try to understand the backbone of 3 dash and upside is 5 dash and this is the sugar, pentose sugar and pentose sugar linked with here phosphorus. Okay, and here phosphorus like this. So backbone, this is called backbone. Now pentose sugar and phosphate, this is known as a backbone of the DNA. Backbone of DNA. So in the backbone of DNA, we try to write the bond name. Here this is the hydrogen bond. Glycosidic bond. This is phosphodiester bond. So like that, there are 3 types of bonds are present in the DNA or RNA. We see the question. Nucleosides were the compounds in which nitrogen bases, purines and pyrimidines, okay, conjugated to the pentose sugar. Okay, asking about this bond. Okay, that is known as beta glycosidic bond. Okay, here alpha, beta, gamma are there. Here beta glycosidic bonds are present in the purines to the sugar. Here beta glycosidic bond. Okay, remember that is a beta glycosidic bond. So third option is correct one. Which immunoglobulin class is most effective to opsonization? Opsonin. Immunology also we discussed a lot in the Kumar Institute of Zoology app. Okay, their total immunology topic is present. Total immunology we discussed a lot. In-depth classes are there. HD video qualities. Opsonization means making a antigen too easily susceptible to eatable by the macrophages. That is called opsonin. Opsonin means to digest, to easily engulf, to make it engulf, or to make that antigen recognized by the macrophage is called opsonization. So when our macrophages detect there is a antigen, it must be show some signals. So that to give that signals, one of the antibody go and binds that antigen and gives the signals to that macrophage, the large eating molecules, and immediately they come and eat that complex of antibody and antigen. That is known as opsonization. Here, the simplest type of our abundantly present in our body. So 85% in our body, it is the IgG antibodies present in the blood. And IgA is the dimer form. It is in the dimer form. It is in the monomer form. So 85% is there and circulating in the blood and also crossing the placenta. So about the IgG. And IgE, it causes a hypersensitivity. This is the answer. So here, in the previously one question is the second question is there. See IgE, hypersensitivity. Hypersensitivity type 1, type 2, type 3, type 4 are there. So just go through the definitions, at least single or double line definitions are there. On the definitions they are asking in the MSc Zoology or in the recruitment examinations about that hypersensitivity. So type 1, type 2, type 3, type 4. Type 1 caused by IgE antibodies, type 1. So here answer is IgG antibody, they participate in the opsonization. Okay, we will stop at the 35th question. This is 34. Next question we will stop here. We will discuss in the another video. A laboratory tool to detect the expression of thousands of genes at a time. See, this is one of the biotechnology question once again. A laboratory tool to detect the expression of thousands of genes at a time. Okay, see, thousands of genes are there and they are expressing their protein function, and that expression we see at a time of the say thousands of genes, that is microarray. This is the microarray technique. This is the technique microarray. In this microarray, at a time we can see the thousands of genes expressions. Last question. Crossing over of F1 hybrid. Crossing over of F1 hybrid with recessive parent is called recessive parent. Suppose this is F1 generation and crossing with recessive parent. So this is the F1 generation and this is the recessive parent. So once again we are doing this cross to test. So this is also called a test cross. So recessive parent means test cross. So test cross is the answer. We stop here, and in the next second video we will discuss the remaining questions. There are total 100 questions are there. The time is 90 minutes for MSc entrance. So this is the question depth. Analyze these questions, pattern, depth, in-depth questions are there. Okay, some questions are very simple, and whatever the immunology, biotechnology, animal biotechnology questions are also asking here. So in the immunology, hypersensitivity, autoimmunity, in-depth questions also asked, and we discuss remaining questions in the next video. Okay, thank you. Like, share and subscribe my channel Zoology Online Guru, and also download the app Kumar Institute of Zoology, and we have so many topics on syllabus for JL, DL recruitment and MSc entrance, NEET, EPSET, like that. So many courses are there with high definition quality videos. We uploaded so many videos, and go through those videos and download the app now. Okay, thanks for watching, and stay connected and come regularly at 9 PM to 10 PM 1 hour live session. Don't miss that one, and we are discussing in the live session about all the systems, and that is called physiology topics. So one topic is completed, that is a nutrition, that is also known as a human digestive system completed, and go through those previous videos, and also come and contact within the live session in the Zoology Online Guru. Thank you. Thanks for watching.